stressful conditions and how these adaptations are linked to pathogenesis. In particular, their studies focus on Enterococcus, an antibiotic-resistant microbe responsible for many hospital-acquired infections, as well as E. coli, a potentially deadly foodborne pathogen. Their research uses a suite of genetic and molecular techniques to decipher whether these genes contribute to their stress resistance pathways. Dr. Fozo, welcome. The screen is yours. Feel free to share screen and you can get started.
Great. Thank you, Pete, and thank you to all of the organizers for putting together this symposium and for the invite to talk to you today. So as Pete mentioned, my lab is particularly interested in how bacteria acclimate to changing environments. And one of the things that we're most interested in is how they either prime their physiology or change their physiology in order to deal with these different changing conditions. So just like us, bacteria are going to experience stress within their lives, and they need to cope with that. And so there are a variety of different things that bacteria will do. They have a variety of sensing mechanisms which will allow them to respond to the stress by changing gene transcriptional pathways. There are enzymes themselves within the cell and within the cellular membranes are going to be very sensitive to these changing conditions, so that can alter the biochemical activity as well as lead to various types of feedback inhibition. And for some bacteria, and this is limited to specific groups, they're able to actually undergo a very complicated differentiation pathway leading to the formation of a dormant spore. Now, spores are much better able to survive environmental stresses because various environmental stressors are going to particularly attack metabolically active cells. Now, many bacteria, though, are unable to form spores. However, for all bacteria that have, I think, been investigated so far, they are able to have a subpopulation within them that will form a persister cell. So persister cells were first noted in the early 1940s by Joseph Bigger and some other folks who were looking at how antibiotics could clear bacterial cultures. And what he and others noted was that if you gave a lethal dose of antibiotics, and antibiotics are going to target active processes in bacteria, they could essentially kill the majority of cells. But you always have these few surviving cells that remain. Upon removal of the antibiotic, those cells would actually grow back out again. And if you went and hit them again with the stress, you end up with the same small subpopulation that survives. Now, we've demonstrated that these persister cells are actually phenotypically altered. They aren't true genetic resistant cells. If they were genetic resistance, either by having mutations in their genomes or having acquired additional genetic elements, in that case, the cells that survive the stress, when they grow back out, the entire population would now be able to survive a second round of stress. So we were interested in how persister cell formation occurs. While this has been focused primarily on in the field looking at antibiotic resistance, it's likely that the formation of these subpopulations are going to help with the survival of any type of stress, whether it be biotic or abiotic. And so this became important for us in thinking about one of the organisms we study in the group, and that is pathogenic E. coli 0157H7, or enterohemorrhagic E. coli. So this is a natural colonizer of cattle and other ruminants. And what will happen as cattle shed, the E. coli will come out as they're shedding in very natural process. But what will happen is those E. coli could end up getting stuck on, more so in the past, equipment that is used for processing of ground beef. It will get stuck on udders, and while people will milk cattle and drink unpasteurized milk, you can actually have that contaminated with the organism, as well as you can get a lot of agricultural runoff in which the organisms will be traveling into fields, in particular leafy greens and sprouts. Now, regardless of what product you end up consuming, this will end up colonizing the host and can cause a very serious enteric disease which in some instances can lead to kidney failure or death. Now, when we started thinking about 0157H7, what struck us was the fact that the intestine of a cow is far different than milk, far different than being in your fridge on some leafy greens or some sprouts, and so on. So this organism needs to survive a whole variety of different environmental conditions in order to eventually make it inside the human host where it could cause disease. So we hypothesized that the formation of persister cells are going to be a critical adaptation for this organism in order to let it cause disease. And in particular, we decided to focus in on the type 1 toxin-antitoxin loci as being critical for this formation. So toxin-antitoxin loci are broadly distributed across bacterial species. They are classified into at least 6 different groups. And I'm going to just walk you through what the type 1 group looks like. So in all cases, toxin-antitoxin systems consist of 2 genes. One will be a toxin and one will be the antitoxin. And they're distinguished between their classes based on the nature of the antitoxin, whether it's a protein or regulatory RNA, as well as some of the biochemical characteristics of the toxin. So for type 1 toxins, these are actually very small proteins. They're under 60 amino acids in length generally, and they tend to be highly hydrophobic. And if you artificially overproduce these from plasmids, you can induce cellular stasis and then oftentimes cell death. Now, the activity of the toxin can be prevented by the production of a small RNA antitoxin. And what this antitoxin is actually going to do is form complementary base pairs to the toxin mRNA. And this base pairing is going to either lead to degradation of the toxin mRNA, a physical block in its translation, or both. Okay, so as long as you have antitoxin present, you're not going to be able to make the toxin and have the profound effects that we see. So to kind of break that down further, how a toxin, a type 1 toxin, may help the cell survive stress, is I'm going to show you this model, that very general model we use. So under a non-stressful condition, you're going to have a toxin and antitoxin RNAs present within the cell. However, there will be sufficient antitoxin small RNA present to base pair to that toxin mRNA and end up preventing it from being translated. Now, when the conditions change and you're under, say, some type of stress condition, the levels of the toxin mRNA are going to surpass that of the antitoxin. There are several different mechanisms that this could occur by. In this case, the toxin will escape inhibition from the antitoxin. You're going to produce the toxin protein, and that is going to lead to basically a halt in major cellular processes such as replication, transcription, and translation. That will lead to a complete halt in cell growth as well as a halt in cellular metabolism. If you halt cellular metabolism, you're going to be far better able to survive a variety of different stress conditions because metabolically active cells are going to be more sensitive to a variety of these stressors. Okay, so many years ago, we identified a locus in pathogenic E. coli as well as across all pathogenic E. coli that we named the Zor-Orz locus. So this is the genetic region found in EHEC, and then this is the corresponding region seen in the lab strain MG1655. So clearly their MG1655 has lost this locus upon its domestication. It consists of 4 genes, the 2 toxin genes, Zoro and Zor-P, and their corresponding antitoxin small RNA encoding genes, Orzo and Orz-P. Now, one of the first things we had to do when we identified this as a potential toxin-antitoxin locus was to confirm it. And so we did the traditional mechanism, which is you clone your toxin gene behind an inducible promoter on a plasmid. And in this case, it was behind the arabinose inducible promoter. We then add arabinose. And when we do that to cells, the cells in red are ones that have Zoro being transcribed and eventually translated. Those in blue do not experience arabinose, so the culture is still able to grow. Now, to confirm that this toxicity can be repressed by an antitoxin, what we did was clone the corresponding small RNA gene for Orzo on a compatible plasmid behind the PLAC promoter. In this case, what we do is we add IPTG, And then 30 minutes later, we'll add arabinose. For those cells in which orzo production has been induced, the culture is able to continue to grow. If orzo has not been induced and we have just toxin production, we acquire cell stasis will occur for this culture. Now, depending on the copy number of the plasmid that we use, as well as the amount of the inducing agent, we can either induce stasis and in some cases, vast cell death when we induce production of Zoro. Okay, so Zoro and Orzo is a type 1 toxin-antitoxin system, but is it going to play a role in helping to form persister cells? So what we decided to do was test the major model that is put forth for how type 1 toxins would do this. So in this case, the toxin protein is hydrophobic, so it's believed to be translated, inserts into the membrane to form a small membrane pore or channel. That will lead to depolarization of the cell. Depolarization will result in a loss of ATP. No ATP, you halt all major cellular processes and will enter this dormant state. So what I'm going to do is show you how we've gone through and tested this at each step along the way. So the first thing we needed to test was, does Zoro actually go to the membrane? So I'm showing you the sequence here. We see that it is highly hydrophobic and has a predicted transmembrane domain. So Vikas Bagati in my lab, in order to kind of track where Zoro may go when it's being produced, he inserted a 1X flag tag at the 5' amino terminus of the Zoro coding sequence, and he cloned this all behind the PBAD promoter on a plasmid. And we have some special considerations we can talk about later if people are interested. So we took this plasmid system and he put it into the lab strain of E. coli and performed subcellular localization. So in this top Western panel here, what we're looking at are control proteins, our cytoplasmic, intermembrane, and outer membrane proteins that have all been tagged. What we can see in this lower panel is actually the accumulation of Zoro in the inner membrane. And so Zoro in our hands is always being localized to the inner membrane for E. coli. Further, our collaborator, Fran Barrera, in the biochemistry department here at UT, has demonstrated that Zoro will interact, synthetic Zoro will interact with liposomes and associate within a liposomal membrane as well. Okay, so our first check mark is done. Zoro goes to the membrane, but does it actually lead to membrane depolarization? So Vikas again took an approach of overproducing Zoro from a plasmid-based system. And then what he did was he used a flow cytometry assay with the dieback to monitor membrane depolarization. And so if we go and look at this, one of the things you'll note is that we're looking at a variety of different constructs, and I can explain those more in depth in the Q&A. If we look in black, our empty vector here at time 0, 15, or 30 minutes after induction with a low amount of arabinose, we see that there's a very small population of cells that are experiencing depolarization. However, if we overproduce Zoro from any one of these different constructs, what you can see is a marked increase in the amount of depolarization. Further, if we add an even higher amount of arabinose, what you can see is that within 15 minutes of induction, essentially 100% of the cells are experiencing a depolarized membrane. Okay, so Zoro goes to the membrane, it leads to membrane depolarization. So will this now lead to a reduction in ATP within the cell? So again, Vikas decided to look at this. He used his overproducing plasmid of Zoro, as well as he had a positive control. So work by Gerhard Wagner's group, as well as Borg Beregoff's group, have demonstrated that the type 1 toxin TISB leads to ATP depletion within inside the cell. So Vikas included that as one of his controls. And what we can see here is we're looking at ATP levels using a luciferase-based assay. And so 30 minutes after arabinose production, this is our TISB versus our Zoro, we see that we have relatively high levels of ATP. This is all relative to cellular OD. But the instance we induce Zoro at different concentrations or a positive control, you see this marked decrease in the amount of ATP in the cells. Further, Vikas has actually monitored supernatant to see if ATP levels are increased in the supernatant. So are we getting leakage of ATP outside of the cell? And he does not see any increase over this time frame. Further, work, again, by our collaborator, Fran Barrera, has demonstrated that if he uses calcine in liposomes and applies Zoro to them, he doesn't see a loss of calcine from the liposomes. And calcine is approximately the same size of ATP. So it appears that how Zoro is leading to a loss of membrane polarization and a decrease in ATP is not by simple gross blowing up of the cell. Now, to further kind of reflect that, what Bikas went out and did again was actually look at the morphology of cells when they overproduce this toxin Zoro. So in this case, he is using uninduced cells, and we can see two different fields here, one more zoomed in as well as one more zoomed out, so you get a better image of the population, as well as cells that are overproducing Zoro from that plasmid, and in this case, he's using a saturating level of arabinose. And what we can see is that the morphology isn't really that much different than from cells that are not expressing this toxin. I want to point out that under these conditions that he's using for Zoro production, if I was to plate colony-forming units, I'd essentially be below my limits of detection. So these cells are unable to form viable colonies. When we take them out of culture, they're essentially all dead. However, we're not seeing gross morphological differences. And this would seem to suggest to us that how Zoro is acting at the membrane is by potentially forming a small ion channel. So coming kind of back to our model, Zoro goes to the membrane, we lead to membrane depolarization and a loss of ATP. So now what happens? Does this then lead into a formation of dormancy? So to first examine this, what we actually decided to do was to look at global metabolites in collaboration with Sean Campagna's lab here at the University of Tennessee Chemistry Department. So to do this, we took two approaches. In the first approach, we took that entire Zor-Orz locus. So in this case, I'm taking both the toxin genes and the antitoxin genes and all of the regulatory elements found within that locus, and I'm putting it on the plasmid PBr322. So what we're basically doing is amplifying the copy number of Zor-Orz. Then what we did was we deleted the entire Zor-Orz locus in pathogenic E. coli. And so what we're going to compare in our metabolites are the effects of having multiple copies of Zor-Orz, approximately 20 to 25 per cell, compared to control, versus what happens when we delete the entire locus in EHEC over a wild type strain that's positive. And now when I show you this heat map, I just want you to kind of get an overall feel for what we're looking at. So if we look in column 1, we're looking at the ratio of the metabolites when we have multiple copies of Zor-Orz versus our vector control. And what you can kind of just see, if you just scan real quickly, is a lot of blue. So multiple copies of Zor-Orz, we see a decrease in the levels of quite a few metabolites. This is somewhat the opposite of what we see when we completely delete Zor-Orz in the pathogen EHEC versus the wild type. So if we look at lane 5 here, some of the things that we see are actual increases in particular metabolites. So this would indicate that the copy number of the Zor-Orz can greatly influence metabolite levels and gives forth some support that maybe Zor-Orz is actually leading to the formation of dormancy. Okay, so we have reduced metabolites, but does this actually mean we have a larger persister subpopulation in these cells? So when you actually perform these persister killing assays using antibiotics, what you will typically see is when you add your lethal dose of stress, what we're going to be using is the antibiotic ciprofloxacin because that's traditional in the field. You're going to have a vast amount of killing initially, and then your killing will essentially slow down because you basically kill the bulk of the population in a short time frame. And so you get this traditional biphasic killing. So we went out and we have tested ciprofloxacin killing of a variety of different Zoro-Orz strains that we have, as well as different Zoro overproduction systems. And I'm not going to go through all those results. I'm just going to show you one, and I can explain more in depth later at the Q&A. So what we're looking here is a wild type cell, as well as a cell that's going to have elevated levels of Zoro coming from the chromosome. And what you can see is after ciprofloxacin treatment, we see a massive decrease in cellular number, colony forming units after 1 hour, and then that sort of levels off after 4 hours. But the kicker is we never see anything different between our Zor-Orz overproducing strains or deletion strains compared to wild type. So we're not getting a change in the number of ciprofloxacin persister cells. So BCAS in the lab luckily was not fazed by this result and decided to push it a little bit further. So he decided to employ a tolerance detection test that has been utilized by the Balaban group. And in this case, what they basically do is plate a lawn of bacteria and drop an antibiotic disc onto that lawn. And essentially you're doing a zone of inhibition assay. What you do after 24 hours of growth is you're going to remove the antibiotic disc and replace it with a disc that contains carbon source, in this case, glucose. If you had cells in that original zone that were not dead, but were basically tolerant and just weren't able to actively grow, the removal of the antibiotic and the addition of the carbon source is going to allow those to rise. And so you can kind of monitor if you have tolerant or persistent cells within the population. So unfortunately, in our hands, we had some very variable results with this assay. But one thing that BCAS noted was that he received various differences in the zones of inhibition when he utilized different strains with Zor-Orz. And so he went out and did a broth microdilution MIC assay. So in this case, we're again using our plasmid that has multiple copies of the Zor-Orz locus, and we're comparing that in the lab strain with empty vector. And so what we can see here is that, again, with ciprofloxacin, we see no change in MIC regardless of the copy number of Zor-Orz. Same thing for chloramphenicol. However, when we look at the aminoglycosides he's tested, kanamycin and gentamicin, we see that multiple copies of Zor-Orz leads to a vast increase in the amount of drug needed to inhibit their growth. He's also switched the backbone and used a PAC-YC 184 backbone and notes similar results. He also was able to test in that strain beta-lactam antibiotics such as ampicillin. And in the case of the Zor-Orz locus, there is no alteration in the MIC for ampicillin. BCAS has then gone on to actually look at cellular growth in the presence of kanamycin. And just again to show you vector control in black versus multiple copies of Zor-Orz, what you can see under normal growth conditions, there's no difference in growth between these strains. However, the instant you start increasing your kanamycin concentration, the strain harboring multiple copies of Zor-Orz grows well, the vector control does not. He can do this using minimal media with either, for example, glucose or fructose as carbon sources. And again, we can mimic these effects. So multiple copies of Zor-Orz is a net allowing these strain to grow in the presence of the aminoglycosides. Now, while this was done in the lab strain, he's also looked in EHEC by moving these plasmids into the pathogen. And similar to what we've seen, no difference in growth harboring these plasmids until you begin to add one of the aminoglycosides, either kanamycin in this case, or gentamicin. Growth in the presence of ciprofloxacin, in the presence of ampicillin is no different between these different strains. So we're kind of left with this interesting conundrum in that we're not able to demonstrate that Zor-Orz is causing a broad persister formation in the population. We're not seeing anything basically until we start to look at the aminoglycosides. And in that case, it's not necessarily persister formation, but basically elevating the MIC and allowing the organism to actually grow. So I don't want to use the word resistant, but there's clearly something different here. And this got us thinking about other type 1 toxins. We all have this model as to how they work, but there are some key differences that have been pointed out by others that I think we really need to reflect on. And that is, when you look at the effects, and this is just a short list showing some type 1 toxins that have been characterized, what you note is not all of them have membrane effects. So FST, one of the first type 1 toxins characterized by Keith Weaver, shows significant nucleoid condensation when you overproduce it. And BSRG, which has been characterized quite well by Sabina Brontel's group, shows that that toxin is influencing cell wall synthesis machinery. So I think it remains to be seen if all these toxins act, one, the same way, and two, are they really involved in persister formation? Or perhaps we need to rethink how we're defining persister, and there may be more nuances, and that perhaps these type 1 toxins all have very specific targets, and that is why we're seeing differences and not seeing broad protection from a variety of antibiotics when we overproduce them. So just to kind of summarize, the Zoro type 1 toxin almost fits into the traditional model that we have for how it may be working. We do note that we're likely forming small channels, and that the additional regulatory elements provided in the Zoro mRNA are very important for physiology and phenotypes. We do see that Zor-Orz specifically impacts sensitivity to the aminoglycosides, but not to others, and doesn't seem to be causing this broad range persister cell formation that we would expect. So we are very interested in following up as to why this may be. And just to point out that we want to think more carefully about these other type 1 toxins and look further into them, as they may possess alternative mechanisms for how they act. It's pretty clear from the data so far, but how they may be actually influencing antimicrobial susceptibility as well. So with that, I want to thank obviously all the members of the lab. This is particular Bikas. So the work I showed you today was essentially from him. He was assisted at times by Andrew Fahim, and in the past has worked closely with Jawen, Oana Stoiklescu, and Thomas Nipper. So with that, I'd also like to thank my collaborators, Fran Barrera and Sean Campagna, for their assistance, as well as NIH for money. And with that, I can take any brief questions if we have time. And if not, I will see people in the Q&A.
Excellent. Thank you so much for that fantastic presentation. I really appreciate it. Let's see. We have, take a quick look here, how we're doing. I believe we have time for questions, and I have a few that have come in over the airwaves here, over the internet waves. All right, so here we go. First question for you, Doctor, is have Zoro homologs been found among the Archaea? It's interesting to consider this as an adaptive strategy to combat stress for bugs that can't sporulate.
Absolutely. So unfortunately, no type 1 toxins have actually been found in Archaea, or at least been very well characterized. And the reason for that, I think, is homology. So for example, if you look, these are really small proteins. And a prime example would be one of the type 1 toxins called the IBS. So the IBS sequence, if you were to take it when it was first identified and try to pull it out and blast in another species, you only see it really in E. coli, Shigella, Salmonella. However, work by Cynthia Sharma doing deep sequencing of Helicobacter pylori found IBS homologs. They just, because of some very few amino acid sequence changes, you couldn't pull them out by homology. So most of the type 1 toxins have been identified primarily in gamma proteobacter as well as firmicutes. And so we're somewhat limited in terms of homology searches. I think that most likely they're present in Archaea. I think it's just, people have to go in and kind of do the old fashioned trying to find them in there and seeing how they may behave completely differently as well.
Yeah, understood. And very much agreed, if I share my own opinion. So we have a few more questions. How pervasive is Zoro among bacteria?
It's actually pretty limited from what we can see. So it seems to be primarily limited in the E. coli species and Shigella species. We think we have some homologs in Citrobacter. One of the interesting things, though, is it has been lost for the domesticated lab strains. So there's been some sort of deletion event that's occurred.
Oh, that's pretty wild. All right, more questions coming to you. Let's see. Dr. Fozo, is there a distinction between microbial persistence versus tolerance? If these populations are different, can you speculate as to the importance of Zoro in their phenotypes?
Whew, that's a good one. So between persistence and tolerance, persistence typically means that these cells will survive that higher concentration and will grow out after the fact. The difference between tolerance, it's a little bit more nuanced, and depending on how you want to define it, can kind of go hand in hand with persistence in a way. What we will say is that it may be able to give us some ideas. I think what what our interest is right now is to why we're seeing such a specificity in our phenotypes. If this was just a global metabolic shutdown, I would anticipate that I'd see more the ability to tolerate or persist in other antibiotics far better. And that's not what we're seeing.
Oh, that's very cool. All right. Are there experimental conditions at which Zoro expressing cells displayed growth or colony formation?
Yes. So, and that will be dependent upon how much production you actually have. So even when we overproduce Zoro from, say, a plasmid, as long as we produce it at low enough levels, we can see the ability of some of the cells to grow. What we're trying to do right now is actually look at production of Zoro from the endogenous level, actually from the chromosome under its native control elements. And that's become a little bit more challenging because it appears that this protein is produced at very low levels within the cellular population as a whole. And we're probably getting a lot of stochasticity in the population as to what cells are making it. So that is something, though, that's kind of our white rabbit right now.
That's excellent. Thank you. More questions coming in. How could bacteria grow faster when toxin dominates and halt molecular machines in the presence of stress, parentheses, antibiotics?
Absolutely. That is something that we are not quite too sure how to explain at this point. One of the things I will state is that we are unclear at this point with those multiple copies. When we see this growth, what are we looking at? How much actually protein is being produced in those cells? Is it being produced across the population? And so those are some of the questions that we have. And I don't have a good answer for that at this point. But those are the things that we need to do, is to do those controls and actually confirm that we are getting high enough levels of the toxin. At this point, all we can say confidently is that multiple copies of the locus allow you to grow better under stressful conditions.
Excellent, thank you. Is the deletion strain hypersusceptible to CAN, meaning kanamycin? Wondering what the function of native expression is.
Okay, so in terms of the deletion strain in CAN, BCAS has been looking at that to see if you can find kind of a sweet spot for concentration where we might see some more sensitivity. So that's ongoing work. In terms of function at the chromosomal level, one of the things, and I can deal more with this in the Q&A and show you some old data. What I anticipate is that very low amounts of Zoro, even though we might have RNA for it, I think very low amounts of it are ever actually translated. And we do have some data I can share later that I think that supports this. So what I anticipate is that probably in a population, very few cells are making the protein. And so again, this idea of subpopulation is probably likely what's happening natively.
Excellent. Aside from forming a spore, is there any evidence that Zoro could be targeting other membrane proteins?
Oh gosh, yeah, that's, yeah. So that is actually something that we've thought hard about, and we are planning on doing some of those assays to actually see if we know that Zoro in a liposome will interact with itself. So we know that it has the capacity to interact with itself, but we need to do those experiments to see. And this is where it gets tricky with expression of something toxic, to actually see if it's interacting with any other membrane proteins. I suspect for the type 1 toxins on a whole, given their different phenotypes, I suspect that many of them are acting on proteins, either within the membrane or at least through other hydrophobic regions of proteins. So I think that's actually a really good question. And I think that there's a lot that hasn't been explored there.
Yeah, indeed. Let's see. So are the effects caused by Zoro and Orzo the same? Maybe I misremembered, but there appears to be two tox-antitox systems that are being discussed here.
Yes. So in terms of, so there's the Zoro-Orzo system, and in our locus, we also have the Zor-P-Orz-P system. Zor-P and Zoro differ by a single amino acid. And we have demonstrated that production of Zor-P is also toxic to the cell. We demonstrate that Orz-P will be able to relieve that toxicity. Given how similar they are, and there are some technical challenges with the Zor-P that we still don't quite understand, we focus primarily on just the one system for right now, so that we have something straightforward. But they only differ by a serine versus threonine in one of the amino acid positions.
Excellent. And our last question for now, until your breakout session is increase in resistance to aminoglycosides due to a collapse of proton motive force, which is required for their import?
So that is one of the things that we have been considering. So we know that we do see a decrease in membrane, we know we see an increase in membrane depolarization. We also see that there's an ATP loss. So this could be. What I am surprised though by is that we're not seeing the effect with other types of antibiotics that we tackled. And thinking that perhaps why not more broadly? Because for some of those other ones, I would have thought that proton motive force would have been important for it. So we're still working on that question a little bit more to kind of get a better handle as to what's happening. But
Yeah.
absolutely, I think it's a very important point that we need to spend more time on.
Yeah, indeed, lots of cool questions. And for those of you who typed in questions into the YouTube chat, we're going to try and capture them and ask Dr. Fozo to address them during the breakout session. Thank you very much, Doctor. We're going to move on now, and my co-host, Dr. Hannah Gavin, will be taking you through an introduction to our science artists. And now our science artists are not with us here live, but please, if you have questions for them, feel free to put those in the YouTube live chat and we'll go ahead and pass those along. Also, if you check our website, you'll be able to find more information about their efforts and their website.